So we just finished our photosynthesis quiz in the College Board app. Today we're going to talk about oxidation reduction reactions. And then as your follow-up, you're going to start reading about cellular respiration in an overview. 8.1 will cover an overview of that, and then 8.2 will get into more detail of the mitochondria, because we really need to understand that anatomy like we did with chloroplast photosynthesis. We need to understand that anatomy, and then that'll help us talk about glycolysis and other and the reactions that follow. So 8.1 and 8.2, I want you to read, take notes. Remember, in AP, it's not about defining words, it's about recognizing how things work. Scientific principles need to be able to be explained, make connections between other concepts, okay? So as you're reading that, examples are good to know. So then at the end of each section, there's like 3 questions, a progress check. So in addition to your notes, I would like at the end of each section, those progress check questions. Do you have any questions on the agenda? That should be a fairly easy weekend for us. So now I want to switch. Okay, so as we're talking about cellular respiration, we're going to get a good handle on oxidation and reduction. So we're going to learn how that process is going to make ATP. That's the big idea of cellular respiration, is that in the mitochondria, we can make lots of ATP, which we know is the cell's energy currency. So that's what we're going to use to make energy for our bodies. So energy is stored in organic molecules. We're used to talking about it being stored in carbohydrates. We want to broaden our understanding of that to the fact that it's also stored in fats and proteins. And we can break any of those molecules down to produce energy. So in 9th grade, photosynthesis made glucose. Now we learn it can make many things, right? And then we learned that cellular respiration breaks down glucose. And now we're going to learn it can break down many things. So you can enter any of these processes at different steps, and that allows you to break down a glycerol or a fat or a protein or even a nucleic acid. So heterotrophs like us, this should be all review right now. Heterotrophs use organic molecules from the food that they eat, they digest it, and they release the nutrients, the raw materials, in order to synthesize other things. So we're breaking down molecules not just for energy, but also to use the pieces and parts to make other things. So we're using energy to control different chemical reactions, right? So we talked about many metabolic pathways that include enzymes, and they have to overcome a certain amount of energy for that reaction to occur. So for the most part, this unit will be about breaking down glucose through catabolism. Remember, you have 2 different types of reactions, anabolism and catabolism. Catabolism is the breakdown. Anabolism is the build up. So the breakdown of glucose is going to allow us to produce ATP. And I see my font fell off of the line here, but the overall reaction, glucose plus oxygen yields ATP energy plus water and CO2. And remember, all energy conversions are not efficient. We lose a little bit of that energy to heat. And I think you might have seen that in one of your questions today on your quiz. So combustion is going to take a lot of energy, like oxygen fuels the burning of fire. Oxygen will fuel our burning of fuels as well, with the help of enzymes. So again, anything that affects enzymes will affect this process. So we're going to digest a large molecules into smaller ones, like, remember, polysaccharides, like starch, glycogen can be broken down into monosaccharides. Polypeptides can be broken down into individual amino acids or dipeptides. Fatty acids can be, or triglycerides can be broken down into glycerol and triglycerides. So whenever we break a bond, we're going to release energy, right? And so this process is going to be moving the electrons, basically, that were in that bond between reactions. And so we saw a little bit of that last week with NADPH and ATP. All right, so when you break a bond, that electron becomes available to be used by another reaction. So when we break that bond and we release that electron, that's considered oxidation. The molecule that gains that electron is reduction. So we talked about NADP plus being reduced into NADPH when it gained the electron at the end of the electron transport chain. So we're going to have the similar process happening during cellular respiration, so it's important that we know what's happening. So oxidation is losing electrons, LEO, lose electrons, oxidized. Gaining electrons is reduced, GER. Leo the lion says GER, gain electrons, reduced. We saw after carbon fixation, you had a reduction of PG3 or PGA into PGAL. So that is making connections with things we've done already. So as we move electrons through electron transport chain, we're also moving hydrogen ions, right? So you can think of losing electrons or losing hydrogen, gaining electrons or gaining hydrogen. Either way, the concept works. So the electrons don't move alone, they move via the hydrogen ion. And so where that hydrogen ion goes, so does the electron. So in this case, we're going to move electrons from glucose into CO2. We're oxidizing because we're breaking the bonds. So this one is losing the electrons. So that's oxidizing. This one is picking up the electrons. So it's being reduced. So coupled reactions, right? Just like we coupled endergonic and exergonic reactions, you're coupling an oxidation with a reduction. We call them redox reactions. So the reduction and the oxidation are in one word, reduction. So as you release energy, by breaking bonds, you strip off electrons. Those electrons will move from one molecule to another. So the hydrogen will shuttle basically the electron between molecules. So in this case, we're oxidizing food. In photosynthesis, we reduced CO2. So the coupled reactions are used and they release energy to synthesize ATP. So here's some rules you can keep straight. We already talked about oxidation as losing electrons or losing hydrogens. You also want to connect it with adding oxygen. And that makes sense. If you add oxygen, it's oxidized, right? And exergonic reactions. So usually if you're breaking bonds, you're releasing energy, so it's going to be exergonic. Reduction, removing oxygen or adding hydrogen and electrons. So I think of this as the charge. So if I'm adding electrons, I'm adding a negative charge, so it's being reduced. Again, you're storing bonds because you're adding the electrons and the electrons are the bonds. So that's storing energy. So if you're adding energy, you're climbing the energy hill, and that is an endergonic reaction. Last chapter, we talked about NADPH. This time we're going to talk about NAD. So be careful to keep the P with photosynthesis, right? P for photosynthesis. Photosynthesis starts with P. So NADH gets commonly confused with NADPH, right? So nicotinamine, adenine, dinucleotide. So NAD is going to be reduced to NADH. It's going to shuttle electrons between reactions. FAD is going to be reduced to FADH2. And then that's flavonoid adenine dinucleotide. And then that is also going to shuttle electrons between reactions. And then it's cyclic, just like NADP was cyclic. You made NADPH, NAD, NADPH, NAD. So we're going to reduce and oxidize so that you can recycle that molecule. So when we talk about cellular respiration, there's 4 main steps. Glycolysis is the breakdown of glucose. That's what the name says, splitting sugar. Aerobic respiration continues if oxygen is available, and you will go through the Krebs cycle. That's going to require a conversion. So pyruvate that is produced during glycolysis will be oxidized into acetyl-CoA. That enters the Krebs cycle, which then produces electrons for the electron transport chain. The end product, ATP, just like the end product of the first electron transport for photosynthesis, produced ATP. You're going to find a lot of commonalities between the 2 reactions, between photosynthesis and cellular respiration. So as you are learning this process, try to identify how they're alike and how they're different. It's like they have the same steps, only the steps are backwards. So look for that all along the way. So in the end, we get to make lots of ATP using that same ATP synthase, that same concept of the hydrogen ion gradient. Okay, so we need to build up the gradient with pumps, the purpose of the electron transport chain. And then when you have a high gradient, it's able to diffuse through the the ATP synthase to combine inorganic phosphates with ADP to make ATP. So this is the same process as the electron transport or the ATP synthesis in photosynthesis. So we're going to call this chemiosmosis. Chemiosmosis. The only different, and this is where I said a lot of things are backwards for cellular respiration. Before we were pumping hydrogen into the thylakoid and it diffused out. This time we're pumping hydrogen out and it's going to diffuse in. So that's your opposite, your same but opposite. And we'll get into that later. Okay, so just a general overview today. Monday, we're going to get into glycolysis, and we're going to cover that in greater detail, similar to covering the Kelvin cycle, because that PGA is going to be right in there again, okay? So today, look for commonalities, look for how they're alike, how they're different, try to understand processes. Today's reading, the first section isn't a lot of detail, it's a lot of importance, and we want to know the importance of everything that's happening inside of cells, right? So that's AP biology. And then the second section gets into the glycolysis, and for you to grasp what I'm saying on Monday, because I'm going to go through lots of steps, it's better for you to read it and get a primary understanding of it before I start talking fast. Today I talked very slow. What questions do you have for me?